Greetings fellow scientists and board members of the Grant Committee. I'm sincerely apologetic for my absence at this prestigious event as I'm snowed in Ulaanbaatar, Mongolia. However, I have a short video explaining my proposed biotechnology and its use in treating Parkinson's disease. I hope you enjoy. Parkinson's disease is a progressive neurological condition resulting in a decline in control over body movements. It occurs when the nerve cells in the substantia nigra of the brain die or become impaired. These cells produce dopamine, which is needed to transmit signals within the brain to produce smooth physical movements. Symptoms of Parkinson's disease appear after approximately 70% of dopamine-producing cells cease to function, resulting in tremors or shaking, loss of smell, stiffness in walking or moving, masked or blank expression, stooping or hunching over, and impaired speech. Most cases of Parkinson's disease are sporadic with no obvious genetic link. However, approximately 15% have a family history of the condition for which several genetic changes or mutations have been identified and a candidate gene has been mapped to chromosome 4. The protein product of this gene is known as alpha-synuclein, also a known constituent of Alzheimer's disease plaques. Mutations in the LRRK2, PARK7, PINK1, PRKN, or SNCA gene have been linked to several Parkinson's disease families. It is not fully understood how these genetic mutations cause or influence the risk of this disorder. Some gene mutations act to interrupt the cell system that degrades superfluous proteins and dopamine-producing neurons. This causes undegraded proteins to gather, resulting in the death of these cells. Other mutations may affect the function of the mitochondria, producing free radicals that can damage these cells. The inheritance pattern of familiar cases depends on the gene that is altered. If the LRRK2 or SNCA gene is involved, an autosomal dominant pattern is present. This means one copy of an altered gene in each cell is needed to cause Parkinson's disease, with an affected person having one parent with the condition. This is exhibited in diagram A, where every affected person in the pedigree has one affected parent. An affected person is shown every generation, and an affected individual transmits the mutation to half or his or her children. If the PARK7, PINK1, or PRKN gene is involved, an autosomal recessive pattern is present. This requires two copies of the gene in each cell to be altered to produce the disorder or affected. The parents of an individual with autosomal recessive Parkinson's disease each carry one copy of the altered gene, but do not show signs or symptoms of the disorder. This is shown in diagram B, where the affected individuals have two parents who are heterozygous carriers, both unaffected. There is a 25% chance that offspring will inherit both altered genes and be affected. If the affected individual procreates with a non-carrier, then their children will be heterozygous and unaffected carriers. Parkinson's disease is diagnosed clinically from symptoms and signs. At a minimum, there must be slowness of movement, bradykinesia, plus either tremor or rigidity. There must also be beneficial response to dopaminergic therapy. Currently, Parkinson's disease is treated with drugs and surgery. The major drugs include levodopa, dopamine agonists, monoamine oxidase B inhibitors, anticholinergic agents, amantadine and catechol-O-methyltransferase inhibitors. For certain indications, surgery is considered, including thalamotomy, pallidotomy, and deep brain stimulation of the globus pallidum pars interna and subthematic nucleus. The limitations of chronic medication use are dyskinesia, motor fluctuations and hallucinations, as well as nausea, vomiting, fatigue, and dizziness. Surgery can result in severe bleeding and infection and does not help all features of the disorder. Given the limitations of current treatments, my work with Prana Biotechnology proposes a new type of treatment. We have developed the PBT434 molecule, which has been designed to constrain the neurotoxic rise of alpha-synuclein in the brain in order to prevent the death of substantia nigra cells as found in Parkinson's disease. In the substantia nigra iron, dopamine and alpha-synuclein protein are all present at high concentrations. In Parkinson's disease, iron binds to dopamine creating toxic free radicals. Iron also binds to alpha-synuclein causing it to aggregate. PBT434 prevents alpha-synuclein from aggregating and prevents the toxic consequences of iron combining with dopamine. In 3 different animal models of Parkinson's disease, PBT434 has proven effective in preventing neuronal loss and preserving motor function. Funding for research into this condition is important as it's the second most common neurodegenerative condition after Alzheimer's disease. The economic burden of Parkinson's disease is estimated at $14.4 billion a year in the US, compared with Alzheimer's disease at $100 billion per year. Knowledge of genetic functions in Parkinson's disease also helps us understand other genetic neurodegenerative conditions with Parkinsonian features like progressive supranuclear palsy and Alzheimer's disease. I urge you to support my quest to find a cure for this terribly debilitating condition.